methylating agents.
Good afternoon, everyone. Today, I would like to briefly introduce the DNA methylation marker, which is newly developed, which hold a great promising to monitor the patient's response for DNA demethylation agent. First, I would like to briefly introduce the concept of DNA methylation. As the graph showing here, DNA methylation is aiding a methyl group on the cytosine of DNA nucleotides, and this kind of modification's major function is to lock the gene in the off position. Accumulated data is showing that DNA methylation operations will drive tumorigenesis. Since such modifications are heritable and reversible changes, researchers have developed a group of DNA demethylation agent which can remove the methyl group from the CpG sites of the genome and automatically cure cancer. Azacitidine and decitabine are two well-known demethylation agents. Both of them has been approved by FDA for the standard therapy of myelodysplastic syndrome. With the support of the Stand Up to Cancer project, and the application of this kind of drug has been extended to solid tumor, such as lung cancer and ovarian cancer. As this graph showing here, this kind of drug has captured several highlights in the most popular press. Well, the faster growing of DNA demethylation drug, researchers find it's important to develop a DNA methylation marker which can monitor the clinical efficacy of such kind of drug. Line 1, which is a repetitive sequence located in the genome, has been first developed by Dr. Jean-Pierre Issa in 2004 as this kind of marker. And this marker, line 1, has been widely used currently. However, in 2009, a report from Blood Journal showed that the line 1 demethylation is not maintained over time. As this bar graph showing here, after 7 days of continued treatment with azacitidine, the patient only showed a 3% of DNA methylation change at day 15 of treatment cycle. And at first day of next treatment cycle, there is no significant DNA methylation change compared to the previous treatment cycle. Such transient methylation change makes it difficult for clinical practice to accurately capture the methylation change after drug treatment. And until now, there is no clear correlation between line 1 demethylation level with clinical outcome. And in our lab, we are currently conducting an interesting project to study the prolonged effect of DNA demethylation drug. Interestingly, we find that of the demethylated CpG sites after drug treatment, only 10% of the CpG sites will regain methylation very fast. And unfortunately, line 1, this marker behaves similar as this 10% CpG sites, which regain methylation very fast, and this result is consistent with the clinical result I just showed in previous slide. And on the other hand, the vast majority of the CpG sites we test will regain methylation very slowly and make this character make this group of CpG sites a best candidate for us to develop a new marker which can be used independently or in conjunction with line 1 to get a more comprehensive and accurate view of demethylation drug's effect. And based on the result I just showed you before, we set up 3 criteria to identify a new DNA methylation marker. So the first one, this markers should show a prolonged demethylation pattern. And the second, this marker must be methylated in both normal and tumor tissue, which is consistent with the line 1 marker. And the third, we would like the new marker can be detectable in both urine and blood sample. And follow this working flow, we collect some normal and tumor sample, and we ran a genome-wide 27K DNA methylation array, and from the bioinformatics analysis result, we finally identified 2 new DNA methylation markers. We call it TH and the DNAI2. As the line graph showing here, in the cell line model, comparing to line 1 marker, our 2 new markers can sensitively detect the DNA methylation change. And as a proof of concept, our 2 new markers can show a sustained DNA demethylation effect even 80 days after drug treatment. And in addition, in 9 MDS patients' urine sample, we find that line 1, there is no significant DNA methylation change after 1 cycle of azacitidine treatment. However, in our 2 new markers, we can detect a consistently and a significant DNA methylation change after 1 cycle of treatment. Although due to the limit of the patient sample number, we cannot show the correlation between the demethylation level and the patient's response, but these 2 new markers still hold a great promise in future, especially in the application in the solid cancer clinical trial. And thank you for everyone. Thank you for the team support me, especially Dr. Peter Jones and the Stand Up to Cancer Project.
Thank you, Xiaojing. This is a complex abstract, as you may have noticed. People are looking for biomarkers of activity, especially with drugs that don't have a striking anti-tumor activity. If you have shrinkage of the tumor or your tumor disappears, then you really don't need a biomarker, and that is the case of GIST treated with imatinib. But in this case, using demethylating agents, the story is different, with the exception of myelodysplastic syndrome, where you really can look in the blood if the abnormal cells disappear, and you can quantify what is happening. When you move to solid tumors, you really would need something that would help you identify whether the drug is stabilizing the tumor, for instance, is doing something to the biology of the tumor that you cannot readily see by tumor shrinkage. So the identification of markers that you could pick up potentially in the tumor through biopsies or maybe even in liquid substances like blood and urine is important. And I think it's important that one has to move on from markers that are not good markers. And we have seen here very clearly that line 1 does not appear to be a good marker to follow for this type of treatment. But they have identified 2 new markers through a very broad and extensive analysis of methylation markers. And these seem to, at least so far in what we have seen in the presentation today, seem to pan out in a relatively small experiment. I think this is a promising piece of data, and it will be even more important when we see now these drugs, these demethylating agents, being more widely applied to solid tumors. And there are now a lot of clinical studies that are ongoing in many solid tumors. And hopefully these, probably not as single agents, but in combination with other drugs, may have a role in the treatment of several of these solid tumors. And in trying to understand what is happening when you apply these treatments to the tumor, we will have to have biological markers trying to help us identify whether we are doing any good to the tumor, in fact, not to the tumor, to the patient. Having said that, then I'd like to